Alright, so what I'm going to talk about today is epithelial mesenchymal transition. And this is an important step in cellular biology where the cells will change from an epithelial phenotype. They'll have a change in cellular molecular properties, and they'll gain a mesenchymal phenotype. And so I'll talk about the phenotypic characteristics that happen during this transition, as well as the signaling pathways that are involved and regulated. And when does this happen? It's during early development, during wound healing and repair, and it also takes place during cancer metastasis. So when you think of generating more cells, you think maybe progenitor. This is important during development. This is the process where different cell types are able to become generated and gain their identity to their final organ identity that they will become, as well as where they're located in the body. You've probably already seen Dr. Gallicano's lecture where he talks about the development and the polarity that happens in the blastocyst and the cell migration that occurs in order to determine what is literally heads or tails in the cell. So when we talk about epithelial cells, these are the type of cells that form layers or sheets of cells, and those are held together by cell-to-cell junctions, and they're important parts of the actin cytoskeleton that are involved in these junctions. They also have what's called apical basolateral polarity. So what that means is that at the base of the cell, it will be interacting with a collagen matrix or a basement membrane. And that's what forms an epithelial layer, such as your skin, something that's more impenetrable, as well as it will have this apical surface, which is able to interact with the environment around it. And they have very tissue-specific functions. So the different organs in the body have different epithelial cells with different epithelial functions. Although overall they serve as a protective sheet in the organ, they'll also have different functions, either pulling things out of the environment, like in the gut, or providing a slick surface, or just whatever is required of the organ. In contrast, mesenchymal cells, sorry, my screen totally went blank up here. Let's look at this one. So in contrast, mesenchymal cells are a 3D arrangement of cells, so they're more likely to be found in the interstitial fluid in the cellular environment. So they form important focal contacts in between the cells, and they have important plasticity and migration potential. They're fibroblast-like cells, so you'll see this like spindly reaching out kind of morphology, and they also have supportive role functions. So they are what are responding to maybe inflammatory signals in the body and making the downstream activities that are important. So taking that inflammatory signal, maybe inducing fibrosis or signaling to another cell that you need to undergo epithelial mesenchymal transition. So also, if you've grown these in culture, it makes sense. So an epithelial cell layer, maybe you've grown them too long and they form contact inhibition and they just peel off the bottom of your dish in like a sheet. Your mesenchymal cells, you often have to grow them in a collagen matrix in order for them to be happy. So some of the characteristics that take place, they're detailed in this paper, this Caluri paper, and even though I'm not requiring any kind of paper to be read for this particular lecture, if there is more background information you want to learn, this is the best paper to go to for that. So when we look at loss of epithelial characteristics, a lot of the markers are shown below that first image. So loss of E-cadherin, loss of lamellin, and then as they undergo this transition, there's change in cell architecture and behavior, and they gain the mesenchymal cell properties, and then properties of those are listed below the green colored cells. And among those in the list, there's different transcription factors, cell surface proteins, the cytoskeletal proteins, extracellular matrix degrading enzymes, which we've probably talked about in previous lectures, the matrix metalloproteinases, the MMPs, as well as downregulation of microRNA expression or upregulation, depending on which way you're going. You can go through epithelial mesenchymal transition, but also less well studied and less less common is mesenchymal to epithelial transition the other way. So to not confuse you, there's EMT and there's MET. I'm mostly talking about EMT, but they are related in many ways. So when we talk about the molecular networks that are involved in this regulation, there are several triggers that can cue the downstream processes that lead to epithelial mesenchymal transition. So firstly, that comes from growth factors, such as transforming growth factor beta, EGFR, endocrine growth factor, FGF receptor, fibroblast growth factor, and HGF, which is hepatocyte growth factor. So despite its name, hepatocyte growth factor, it actually also has a role in the formation of other types of organs in the body, particularly during development. So once these outside factors bind their receptors, such as the TGF-beta, it has its own TGF-beta receptor, the GTPase family members, which are downstream, become activated. So you've talked about these before, the Rho, RAS, CDC42, those different family members. So once those become activated, they can't go directly into the EMT program. Instead, they have to go through these regulatory mechanisms. So SANI1 and SANI2, also known as snail and slug, and you'll see those kind of interchangeable throughout the presentation. So just be aware of the change in distinction of what their names are. And you can also see there's extensive crosstalk through the pathways. So this is important for regulation. So although a lot of cell proliferation is good for the body, you want to see that happening during wound repair and development, it can also be bad because it can get out of control, like in cancer. So all of these important checkpoints will help prevent that from happening at the wrong time. These are 4 key elements of epithelial mesenchymal transition. So the first one is the receptors. So what is actually binding the growth factors at the cell surface. So this is a first point of contact. So that something may be afoot in the cellular environment. So the receptors are going to respond to a signal. And following that, the junctions are going to become dissolved. So you can see this cell here is undergoing epithelial mesenchymal transition. So it's received a signal, it's breaking its junctions with the cell next to it, and it's also going to break ties with the basement membrane that it's bound to. So through integrins and induction of extracellular matrix. And then lastly, it's going to gain motility, and this allows it to have the migration potential and the plasticity that is characteristic of a mesenchymal cell. So to detail what happens in the junctions, so again, the growth factors that are binding are shown here, TGF-beta, EGF, FGF, HGF, they're going to bind their specific receptor. They're going to undergo regulation through snail1, snail2, SANI1, SANI2, snail, slug, all the same thing. And twist, which is also another transcriptional regulator. And this will inhibit genes which induce the expression of E-cadherin, and it will also, and in contrast, it will induce the formation of matrix metalloproteinases. And in this second panel is kind of more detail of that. So this is your E-cadherin, and it has this important beta-catenin at the base of it, and that binds the components that lead to the actin cytoskeleton structural characteristics. So when you think of this E-cadherin structure being degraded, it's kind of like thinking of like a hot air balloon. It's being held by all these ropes. You cut the ropes and it's going to be set free. It's no longer going to be constrained to the shape that it's being held into. Okay, so this details the matrix metalloproteinases moving in, degrading that E-cadherin, and there's also downstream interaction with, so your growth factor receptor is binding, your E-cadherin is being degraded. There is phosphorylated SARC and phosphorylated FAC, which move their phosphate to the C-terminal end of E-cadherin, and also phosphorylation of beta-catenin. And what this does is, through this ubiquitin ligase, there's degradation of the C-terminal end of E-cadherin, and then there's also phosphorylated beta-catenin, and that moves to the nucleus, and that will help initiate these pro-invasive genes. So on the last slide is one way to regulate ecadherin, which is kind of shown all the way here on the right. And it can also be regulated through these different transcriptional regulators, BHLH and zinc finger factors. So you've probably heard of zinc fingers before, important for transcriptional regulation. And then these bind specific sequences on the DNA and are able to affect transcription of the different genes that are needed for epithelial mesenchymal transition. BHLH, which I discussed. So thirdly, there's this increase in collagen 1. So this enrichment takes place maybe during a disease state or during development or cell senescence. So this is the collagen 1 matrix, which again is the basement membrane part of the epithelial cell that's important for the polarity that is seen in the cells. The important thing here is the upregulation of epithelial. There's a lot of molecular factors, these different receptors, and again the phosphorylation and the downstream effect on the filaments. But the important thing to understand, you don't even have to remember all these little tiny molecules because nobody's really got the energy for that when you don't have to. So I'm just going to say that upregulation of epithelial is the most important part of this, and there are many related signaling pathways that lead to its dislocation from the basement membrane. Okay, so lastly, membrane protrusion. So this is what helps the cell gain its motility potential. Your platelet-derived growth factor receptor here interacts with the epithelial on the cell surface, and again through downstream mechanisms, this phosphorylated tyrosine phosphatase. What do phosphatases do? They make sure that the phosphorylation cannot occur on beta-catenin. So you can see this inhibition here, and we just saw in the previous slide that the phosphorylation of beta-catenin is important in order to move it to the nucleus where it can begin transcription of these genes. So by inhibiting that, it's inhibiting that process, and again moving into a cell motility environment where the cell can begin to assemble this actin cytoskeleton and basically be able to move. It's set free from its cellular constraints. So when we look at the cellular changes that occur during the gaining of these motility properties, there's three major, structural changes that occur. Firstly, the invatopodia, which is this actin-rich protrusion shown here under panel A. And so this possesses extracellular matrix degrading capacity. So again, cutting those ties and reorganizing the structure in order to make it one that is strong enough to move to wherever it is needed. Secondly, lamellipodia forms this network. So I think of it as kind of like a wave. So it's kind of this force that's kind of pushing the cell forward. It's kind of dictating what side of the cell is pushing to whatever direction it needs to be. So this is like the wave of strength inside the cell that's pushing it to the forward. And then the filopodia is kind of this stiffer extension, so maybe like a finger grabbing onto the surface and finding actually where it can bind or adhere in order to move the cell forward. So I've talked about TGF-beta a lot. It's important. TGF-beta through SMAD signaling is an important initiator of the transcription of these transcription factor family members, such as snail. So again, SNI1, SNI2, snail slug, all the same thing. The ZEB family and also BHLH family. And these will either activate or repress target gene transcription. So the regulation, you can see these are all the downregulated genes here in the green, and the upregulated ones shown below. You'll see a lot of the extracellular matrix and cytoskeletal elements that you've seen before in Dr. Gallicano's lectures. I think he talks a lot about the actin, breaking down of being reorganized. So there's like the occludins and collodins, the ecadherins, desmoplakin, placoglobin, all of those, the matrix metal proteinases we talked about, and a lot of other components that are important for the you see them. But again, the ones you've seen most and are familiar with, those are really going to be the ones that I'm going to say are most important for understanding in this lecture. TGF-beta, aside from SMAD signaling, can also have an influence through these other pathways. So through the Wnt pathway and through the delta-jagged pathway. And again, inhibition of beta-catenin, inhibition of E-cadherin, inhibition of V-cadherin during epithelial mesenchymal transition in response to a TGF-beta signal. So you only see this inhibition when the TGF-beta response is initiated. Okay, so looking at these three families of transcription factors, the first one I'm going to talk about in detail is the SNAI1, SNAI2 family members, and this is their structural elements. And their function has been found during developmental processes, but they're also currently under a lot of investigation. So if you look at the papers that occur, or the papers that are recently published, they still are not sure what exactly every part of the transcription factor is important to the different properties that it has. So there's a lot of information that you don't have to know. So here's a serine proline-rich region that allows for the diversity in these transcription factors. And there's NES domain, which helps determine if it's going to be located in the nucleus, or the slug domain, which is also can vary and what am I trying to say? It contributes to the specificity of the SNAI1 and SNAI2 transcription factors. The phosphorylation at this NES box determines where it's going to be located. The E2 box is where it binds on the DNA. This is the particular sequence that it likes to bind to. Again, for the love of humanity, do not memorize that sequence. It's not worth your time. But what you do need to know is that it's a zig-figure transcription factor. Very simple. And it's diverse. There's a family of them. So the ZEB family, so there's two members, ZEB1, ZEB2. These are zig-finger clusters, and the structural components that are important are at the N and C terminal ends. And this binds to the Z box on DNA. Again, it gives you a specific sequence of where that is. And these act as a transcriptional repressor, and they're activated during development, and also during the other two types of epithelial mesenchymal transition, during fibrosis, wound repair and healing, and during cancer metastasis. BHLH family members. So how do we remember this? It's because of the structure. It is a basic end with a helix, loop helix, BHLH. So it's just simply named after its structure. So this should be easy to remember. There's two alpha helices. These are joined by a loop, which is required for dimerization. If the BHLH family members are not dimerized, they are inactive. So this is important for its regulation. And if it is not dimerized, it is not active. So whatever inhibits its dimerization is inhibiting its transcriptional activity. So classes within the BHLH family members are twist 1 and 2. And these are transcriptional repressors or activators, and they can have tissue-dependent functions. So the transcriptional regulators are, again, themselves regulated. So GSK3 mediates the phosphorylation that can move SNAI1 into the cytoplasm, where it can undergo ubiquitin-mediated proteasome degradation. In previous lecture, I think or maybe upcoming lectures, you talked about proteasomal degradation. So this is this complex here. So depending on whether it's phosphorylated or not, will determine where it is localized in the cell and whether it needs to be degraded. Similarly, SNAI2, ZEB2, and BHLH are regulated. These subsequent slides, so again, if you can't see this very well, you can go to this reference here, and it has, you'll be able to blow it up a little bit in more detail, but you should also have the slides on the Blackboard site. So the ZEB family, so this is ZEB1 or 2, they undergo simulation. So these are small ubiquitin-like molecules that bind the ZEB family members, and those will also help determine its cellular location, so nuclear, cytoplasmic, or will it be moved into proteasomal degradation. Lastly, the BHLH family members, which I just discussed, must be dimerized in order to be active. So this dimerization can be inhibited by its phosphorylation state and also by these other molecules that can bind it, but it's not, again, dimerized with its BHLH friend, so it doesn't have the same activity as it would. So it needs to the BHLH family members need to dimerize in order to have their activity. So these are important regulatory steps to understand. So transcriptional regulators are important for regulation, but they themselves are also regulated by intracellular activities, such as like these that I've shown. So there are specific genes that are targeted by these different family members. So again, there's a lot of excessive information here, but look for the ones that that you've seen before in class or that you've seen in these slides. So the occludins, collagen, extracellular matrix components, smooth muscle, actin is an important extracellular matrix protein. But yeah, all the little ones that seem unfamiliar are unfamiliar for a reason, because I'm not discussing them in relation to EMT, which means you don't have to know them at least for this lecture. Okay, so within epithelial mesenchymal transition, there are three types, so which I've kind of brought up, but now I'm going to go into more detail. So the first type is epithelial mesenchymal transition that occurs during development. So what allows these cells to gain their final identity when the neural crest, the primitive streak form, and they start budding off into their different organs and different cell types. Type 2 occurs during organ fibrosis and tissue repair. So when there is a wound or when there is some kind of chronic damage to an organ, you might get a fibroblast response. So for example, in the liver, I study the liver. In fatty liver disease, there's a lot of fat accumulation in the liver. In response to that, fibroblasts become activated and they release this extracellular matrix. This forms fibrosis, a progressive stage in liver disease. Following fibrosis, you'll see the breakdown of cellular architecture. That's known as cirrhosis. And so with each progressive stage of the cellular response, you also can get increased damage. So, but also this can just be a simple wound, like when you cut your skin, your fibroblasts are being activated, they're moving in and they're forming like a scar tissue. So this can happen in any of those situations. And lastly, the one that we might be interested in studying, but we don't want it to happen to us or our loved ones, is EMT associated with tumor metastasis. So this is where maybe a tumor lesion is not spreading, maybe it's fairly benign, and then different EMT transitions occur, and it can suddenly gain this potential to invade and metastasize the tumor and change the identity of the cells in such a way that it can take over. Okay, so within the three types, type 1, type 2, type 3, type 1 has primary, secondary, and tertiary EMT. So I'm going to talk about primary, secondary, tertiary, but I don't mean type 1, type 2, type 3. So make sure you understand that distinction, because because you'll get lost, and I would be really sad to see you get stuck in this. In gastrulation and amniotes, there's this first EMT event at gastrulation, which the cells of the apoblast move and become part of this primitive streak. So your primitive streak, again, is the source of where your progenitor cells come from. You'll see the budding off and the identity of these cells change as they gain their progenitor-like function for their liver, the heart, the lung, whatever it is that their destiny is. And this helps form the three germ layers. So they move in and they form the mesoderm and the endoderm, and they move inward. So this is this is a primary EMT. So this is the very basic level at which these cells gain a different function or role in their identity. Is the very first step of development. And so this pathway is tightly controlled during development, because again, if anything goes wrong, you don't have life. It's not going to develop properly. So this is very tightly regulated through the transcription factors. We talked about the transcriptional regulators we talked about before. Again, you see snail in here, and there's other ones too, which I'm not going to highlight. Instead, I'm going to talk about the snail, and we've seen the downstream gain of mesenchymal markers, the loss of cell adhesion and polarity, and the ability for it to gain invasive potential and become and gain its identity. So, related to this EMT and the this primary EMT, the important thing to understand is that it's gaining its identity. So, through this pathway, it can gain its endoderm function. Through this pathway, it can gain its mesoderm function. So you're forming these 3 germ layers. But all of it is through EMT. Forming the 3 different germ layers and how it's regulated. So the primary EMTs, which give rise to organ progenitors, undergo a secondary EMT. So primary EMT, you're forming the germ layers. The germ layers gain their specificity through the secondary EMT. So they're mesenchymal cells now with a more restricted differentiation potential. So now it's going to become part of the intermediate mesoderm, the paraxial mesoderm, the lateral plate mesoderm. So it's still very general, but specific progenitor type identity at this point. So this also occurs during neural crest formation. Again, the function of this is, it's still under investigation. So what they do know, though, is that there's this delamination which occurs during neural crest formation, and this is an epithelial mesenchymal transition that allows the neural crest to separate from the neural tube and become the different cell types shown here with the different identities that they will become. So again, with progressive rounds of EMT, specificity increases. I mean, that's the take-home message. So neural crest formation, also tightly controlled. I can only say it so many times, snail 1 and 2, important regulators. Downstream effects for, in this case, neural crest delamination, but EMT specificity. You see the bone morphogenic protein and fibroblast growth factor are ones that you might recognize also in related pathways. So tertiary development, so for example in the heart, a very complex organ, this undergoes an epithelial mesenchymal transition initially, and then it undergoes the opposite, the MET, the mesenchymal to epithelial transition. And what this does is it forms 2 epithelial layers in the heart. So this is the tube during heart development. And then, if you remember from your development lecture, this tube kind of folds over on itself, and then making a crazy balloon animal, it's able to reorganize and produce the 4 chambers of the heart. So this is a very complex process. So primary EMT, cell identity, secondary EMT, you're forming the layers, and then tertiary, you're actually forming these complex structures, such as the heart chambers. So there's lining of the heart chambers, there's different muscles, it's a very complex organ. Okay, and it's also important during gastrulation. So Drosophila, for example, they have slightly different developmental, method of development. Once these cells ingress, so again you have kind of your original blastoderm, you have cells that are going to ingress and gain the specificity that are going to become a final organ or structure through EMT. And these undergo rapid changes, which are again regulated by snail, and in this case twist, which is a BHLH family member also, which induce the different EMT related genes. So now I'm going to talk about pathological EMT. So this is type 2. So that was all type 1 development. And there's primary, secondary, tertiary within type 1. In type 2, there's none of that. So it's just type 2. Very simple. Well, not very simple, but more simple. What happens during type 2, again, is, so this is the response to tissue damage, to injury. This is going to form the scarring and the release of extracellular matrix and activation of fibroblast. So what occurs here is your fibroblast is hanging out in this extracellular matrix in a tumor microenvironment or within an organ in the body. and it has receptors and integrins and different components that interact with the cellular environment. Once it receives a signal, so maybe inflammation, so for example, again in the liver, so when there's a lot of fat accumulation in the hepatocyte, the liver cell, they're going to release all these inflammatory factors that will be received by the fibroblast, activate it, which allows it to gain these motility functions and release the extracellular matrix that is going to be a response to the inflammation or to the damage or whatever the situation is in that particular organ. Let's see what else is it. Produce that extracellular matrix, yeah. Production of alpha smooth cell actin, so this is an important marker, and if you study fibroblast activation in your labs, this is one that you'll use in your Western blots or your staining or whatever it is to show that your fibroblasts are in fact activated. They're not going to release it if they're kind of in this quiescent stage. Once they're activated, alpha smooth cell actin is a perfect marker because it's an extracellular matrix protein, which is one of the components that a fibroblast releases once it becomes activated. So in tumor stroma, fibroblast is called a cancer-associated fibroblast when it gains these roles because of the tumor microenvironment around it. And this cancer-associated fibroblast is very important because it can release interleukins in response to inflammation, and so it's kind of, they work together. Inflammation induces the fibroblast, the cancer-associated fibroblast, and again produces the interleukins that are going to initiate this inflammatory process. So you kind of see how this can get out of control fast if it's not properly regulated. It also exchanges transforming growth factor beta with cancer cells. So the growth factor initiates growth in epithelial mesenchymal transition. So again, this is going to be a way where cancer-associated fibroblasts can induce metastasis in, maybe a benign tumor lesion or cancer cells that maybe don't have an identity yet, which is going to induce an immune response in the body. It can also, these cancer-associated fibroblasts, through the growth factors, again, they can take these perfectly nice epithelial cells that are living their life and doing exactly what they're supposed to do, and it can make them change in response to these growth factors. Maybe they'll think, oh, you know what, now I'm being bombarded by these growth factors, I need to change my identity, and maybe I'm going to leave a hole here which an invasive cell can penetrate and just wreak havoc in the body. Also, it can work through VEGF signaling to induce angiogenesis. So in the tumor microenvironment, hypoxia, so the loss of oxygen, is a major issue. So it tries to induce angiogenesis in order to grow. So it needs oxygen to grow. So it's going to induce blood vessel formation. So similarly, the cancer-associated fibroblasts will send signals that allow that vascularization to occur, which is also bad too, because you don't want to have a blood vessel infiltrating your tumor because you're going to get a higher chance of metastasis. You're going to get a place where the metastasizing cancer cell can penetrate and move throughout the body freely. But it's not all hopeless, okay? There is still regulation that occurs in the tumor microenvironment. So in response to binding of these different growth factors, or the matrix metal proteinases, or the growth factor beta, or the collagen-enriched environment, it can activate the GTPase family members, which again trigger this epithelial mesenchymal transition. So inhibitors at these different stages are important targets for inhibiting this process and inhibiting metastasis, which is detailed here. So like I was talking about, you have this tumor lesion. Epithelial mesenchymal transition can allow these cells to change their identity, sneak in through a blood vessel, and metastasize to a different part of the body. And what's interesting too is if you've read Dr. Schlegel's research or you're familiar with his conditionally reprogrammed cells, there's a similar method for that, which takes the identity of the cell using a Rho kinase inhibitor and allows it to kind of stay in a quiescent state and then regain its identity once the conditions have changed again. So, so this kind of transitory phenotype is important for cell metastasis, and it's induced by epithelial mesenchymal transition. Okay, so similar in disease, I'm going to bring up the liver again because I love the liver. For example, you have normal epithelial cells. They're acting just the way they're supposed to. They're receiving inflammatory factors from the environment. There's a lot of lipids in these cells. We need an inflammatory response. There's some issue. There's cellular damage that's occurring. Then they're in this partial EMT state where they continue to express epithelial markers. So they're still bound to their friends. Maybe they're still bound to their basement membrane, but they're beginning to gain mesenchymal markers. But they're still trying to, escape. So this is kind of a transitory phase. And then eventually they can gain this mesenchymal phenotype, and this will allow the organ structure to degrade in a situation of disease, such as liver disease. Once you've reached cirrhosis, it's irreversible. Just fibrosis, inflammation. So maybe at this stage you can still reverse it by reducing the factors that lead to that kind of inflammation, or inhibiting these different factors. Another example here is in renal fibrosis. So in response to the immune cells, the mononuclear cells passing through the kidneys, the kidney tubular epithelial cells, you'll see this change again. So this epithelial mesenchymal, in between cell, and then this mesenchymal properties, and then disruption of the normal functioning tissue. Like I thought, I went through this kind of fast, but there's not a lot to it other than understanding EMT, what is it, what factors are involved. So we know it's a change in cell identity. There's cellular and molecular factors that are involved as far as what is happening on the cell surface, what is binding the receptors, what are the cell-cell junctions that are being broken down, what are the junctions, where are the integrins, what is happening to them on the basement membrane, what are the different motility factors that are being affected, the cytoskeletal rearrangements and those sorts of things that are involved. The transcriptional regulation through ZEB, SANI, and the BHLH family members. So, what are these guys, basically, and how are they themselves regulated? And also characteristics of the different EMTs. So type 1 during development, type 2 during fibrosis and repair, and lastly, in tumor metastasis. And then also in the slides are the references where a lot of these images were taken from. The one underlined is the one I highlighted in the beginning that is really helpful to understand the basics of the EMT. With that, if there's any questions, I could answer them. If not, it's Friday, and this is not too bad of a lecture, I hope. So any questions, let me know. And also on that first slide is my email. So you can email me later too. I know the exam is like a far way off. You guys just had your last exam. But as exam time gets closer and you have any questions about this, you can email me and just let me know if there's something. Yeah.
Quick question.
Yeah.
I know that EMT is really associated with like invasion and metastasis, but can you still have EMT in tumors without metastasis?
Yes, you can, but the tumor environment that induces EMT it's going to be a microenvironment that is going to favor EMT. So if for some reason you can reduce maybe oxidative stress or different things that are provoking that tumor environment to want to become epithelial mesenchymal transition-like, then I could see that process being limited. But yeah, once that microenvironment is ideal, they're just going to take off.